Morning, everyone. It's really delighted to be here. So a good story always begins with the lines, a long, long time ago. And for bromoform and methane, you've got to go back to actually 1972. That was the first report of an in vivo trial that was reduced methane. It involved sheep, and they reported 100% knockdown with bromoform over 24 hours. Now, as a New Zealander, we hate to give anything to the Australians, but the person involved was J.W. Lanigan, who was working in CSIRO, so Australia. He was a veterinary microbiologist. For much of the next few decades, work on halogenated compounds was sort of confined to the sort of research arena. In fact, even when I started in Rumen Micro back in the early 2000s, we were sort of phasing out the use of halogenated compounds in terms of chloroform and bromochloromethane. And it wasn't until researchers, sorry, Kiwi colleagues in Australia, again identified obviously the red seaweed and its impact on methane emissions. Since then, for many of us, we've sort of watched the science evolve before our own eyes, starting with, of course, red seaweed. They then went on to, of course, identify that it was halogenated compounds that were the active ingredient, and then focusing into bromoform, which was the most active compound. In the last decade, of course, we've had a lot of developers come into the space, which has been amazing to see, and they've driven sort of innovation in this area, starting off with can you feed seaweed, then looking at seaweed extracts, then people, of course, focusing in on synthetic bromoform as well, and also moving the conversation on towards, how could you deliver it? Can you deliver it in a pellet? Can you deliver it in water? Can you deliver it in a bolus? And the story continues still today. And so this is a nice segue into our presenters. We're going to have three presenters. They're going to do three presentations, about 10 minutes, I'm being told. And then we'll go straight into panel Q&A. It's not often that we get this quality of a panel together, so I really encourage you to make use of them. This is a safe space. I'd rather we ask the hard questions here than, for instance, down the line, me having to try to explain to a farmer why we didn't answer those questions in the first place. So I encourage you to use the Q&A Slido, and hopefully we'll have a very good session. And by the end of the three presenters, we'll all be up to speed with where the current research is when it comes to bromoform. So with that, and I'm looking to see if I can see Ermias. Yes. Here he comes. I don't really need to introduce. I'm sure there'll be plenty of references to the FIFA. He's on home soil. So yeah, very welcome, Professor.
Okay, good morning, everybody. All right, so I can see the presentation up here. So it's my pleasure to present today on bromoform-based feed strategies, particularly looking into the bromoform-based feed additives. Last year, I think when we all met here, we were working on this meta-analysis, and so now it's already published. So I'm going to focus on what we found on this meta-analysis, where a lot of the work has been done. As Sinéad mentioned, since the first time it was recognized that bromoform was the active ingredient, there's a number of work that has been done, whether it is seaweed or whether it is synthetic bromoform. I think this research has really increased in volume, and so we decided to put together all the information that we have and try to have a meta-analysis, a systematic analysis, and then understand what are the factors that are affecting emissions or emission reduction from bromoform. Okay, thank you, Charles. Okay, maybe I'll move a little bit closer too. Okay, But basically, when we talk about bromoform, as already mentioned, it's there are a number of things that it will do. It is in some cases naturally occurring, and it's quite selective in which the way that it is producing as well, and it is a volatile compound. So a lot of the times that the issue has been, how do you try to get the bromoform to be contained and be available to the animal, right? So we don't want the bromoform to be released into the atmosphere. It needs to be delivered to the animal so that all the function that need to be done on the animal itself. So we looked into the effectiveness of methanogenesis. So this meta-analysis that we've done is really try to figure out what is the effectiveness and what doses will it be effective at. So we had about 14 in vivo studies that was done, and we compared about 39 treatment comparisons, and we had to put together the beef and dairy, just because we do not have enough data for this to have a separate beef and dairy as we did for 3-NOP, for example. But as we get more and more on data, that's what we're going to be doing as well to see what the effectiveness are. And we've seen some evidence that there is a little bit of difference in terms of effectiveness when you do it with the dairy compared to the beef. So the mean that we've seen was about 28.3 milligram per kilogram, but it's different between dairy and beef. Well, I'm going to show later on what are the averages for beef and dairy, but overall it's about 28.3. And then we have a nice spread in terms of the concentration that was given to the animals as well. So it's spread between 2 milligram per kilogram up to 104 milligram per kilogram of dry matter as well. And then we looked at, as I said, beef and dairy. We looked at all the studies have to report the bromoform concentration. So we need to know what is the bromoform dose. And then we also make sure that there is a direct methane measurement as well, so that we can compare the measurement methods as well. And we did a couple of different analyses. So we did this from the traditional sequentist method, as well as Bayesian analysis as well, to try to make sure that whatever we find is not an artifact of sort of the statistical analysis that we're doing, but it's actually robust, even if we do it in different ways. And then we looked at the production, yield, intensity, looked at the production parameters, the animal performance, and dry matter intake, and then other factors that may impact on the efficacy of the emission as well. So the big headline, I guess, is that at the mean, so that's 28.3 milligrams per kilogram, and also mean concentration of the fiber and the starch and all levels, then we expect about 47% reduction in emissions, about 43% in milk in methane yield, and about 39% in methane intensity. So this is, again, overall. If we separate it out with beef and dairy, it will be a little bit different. But overall, this is the reduction that we saw. And it has a little bit, so the way that it works is that there is a modification of starch and particularly starch and NDF, which is kind of in the inverse proportion. When you have more starch, you have less fiber. And so it's not the same. It's just because of dose, you're not getting that reduction. So the dose is modified by the dietary the nutrient composition of the diet, right? So this is kind of the illustration, which that is not a plane, but it's actually curved, and that is modified by starch in this case. So the dairy, the concentration we have in the dairy is about 15 milligrams, while in beef is about 37 milligrams. So there was quite a big difference in terms of the mean dose, and the intake, the dry matter intake change when you have at this average dose for dairy is about 6% or 6.5%, and for beef was about 3.2% as well. And in terms of the production outcomes, we've seen for dairy about 4.6% reduction in milk yield. But in beef, there was an improvement of about 0.86%. as well, but it was not significant. So at least in this meta-analysis, we did not see a significant improvement in yield. Some individual studies have seen an improvement in yield, particularly in beef cattle, but here in this meta-analysis, we haven't seen a significant difference there. So I just want to take you into a study where we're trying to understand this in a more systematic way. So a lot of the studies that have been done, included in the meta-analysis, were done around 3 to 4 months. So we wanted to know what was the impact when you have it at a longer period of time. At the same time, we also wanted to see if we can use the bromoform, in this case, brominata from Blue Ocean Barns, if we give them at the beginning of life for like 2 months, and then we stop when they are weaned, and then we continue to measure. So can we do something? Can we change the microbiome early through the use of brominata or bromoform, so that the microbiome is changed, and then can it carry over? For how long can it carry over? So this is the work that we've been doing for the last couple of years. And the results are that we've seen some carryover effects. So the blue lines over there is the short term. So the blue line basically means we gave them for 2 months until they are weaned, and then we stop and we continue to measure. While the green line is we started after they have been weaned, and then we continue until the end of life. And so what we've seen so far is when we started from after they are weaned, all the way onto maturity or onto harvest, there was no the animals didn't get used to it or anything like that. So until the last day, we always see a reduction in emissions. So we are about to finish this. I was talking to Paolo, who's doing this work, has been doing this work for the last 2 years, and this month will be the last month for this project. So we're getting this is almost a complete picture, not quite completed, but the effectiveness is there right until the end, which is very much reassuring. And for the ones that we started at the beginning and then we stopped, relative to control, we have also seen some reductions. We've seen an average of about 10% reduction over their lifetime. So it's meaningful, but again, we haven't finalized the data yet. So hopefully we'll get to that and we'll report that soon. And in terms of the performance outcomes, what we've seen is overall, there is no significant difference in milk yield, in average daily gain. But what we've seen is that in the last 2-3 months, there's kind of a shift, that there is an increase in productivity. So again, we haven't finalized this yet, but it's very curious to see that the average daily gain and diameter intake lines were just on top of each other for all this time. And then right at the end, we've seen a bit of a divergence where we see a better average daily gain. So we will delve a little bit more into that and then do some more analysis on that. So basically, it was the end now. I think my time is up. So the evidence tells us that the bromoform containing compounds, they do work. There's a methane mitigation effect, at least about 47% at 28 milligram per kilogram, but that could be different depending on how it's being provided as well. The biggest factor, obviously, is the dose, but it's not just the dose, but the nutrient composition of the diet has an impact on the effectiveness of the bromoform as well. And we've seen some impacts on intake and on yield as well. I think the things on intake are most related to the palatability issue. I think that those kind of things are being solved right now. So I would expect moving forward that kind of issue to be solved, and we won't have as much a reduction in intake. So the only gaps that we have is that looking into the long-term impacts, and this is what we're trying to fill at the moment. Once this project is over, we will analyze everything in terms of the organs, liver, and kidneys. We also look into the meat and all that, making sure that there is no unintended consequences. Thank you.
Okay, thank you. So next up, we're going to the other side of the country. So can I please welcome Dr. Joe McFadden from the Associate Professor of Dairy Cattle Biology from Cornell University. Welcome, Joe.
We'll try talking here. Hopefully you can hear me in the back. Is that good or no? No? Okay, here we go. All right, I'm usually a loud talker. Ermias got 11 minutes, so I get 11. This started, my journey on this path started right around COVID in 2022. I had an op-ed that I wrote that was really critical of seaweed and bromoform. And then once I submitted that in 2022, I was like, yeah, I'm never going to get funded to do bromoform work. That quickly changed. With that, we had some criticism as well. Immediately when we got funding from CDFA, there was a good a blog that was put out by Marion talking about how this was marketing and not science. And she did not read the proposal. She's from Ithaca, never even came to talk to me. Long story short, I invited her to my class and I put her in the chambers. I didn't lock them, but I did put her in there. That said, we had a conversation, and I think it's something to be made aware of, that this kind of topic's going to have a lot of attention moving forward, especially with industry influence. That said, we had a plan, was sponsored in part by CDFA, to sort of do a side-by-side comparison of different bromoform technologies, which I really wanted to do since I sort of started this process. We want to look at bromoform stability, do a sort of a mini pilot study with 50 cows to sort of look at what we might think is the best feeding level to target, and then perform a series of 12-week studies that were more intensive, looking at acute and long-term effects and maybe what's going on in the rumen with these different products. We also want to do an interaction study with bromoform by fatty acids, and then hopefully next year finish with a dose titration study. And we have currently had a budget for 36 necropsies, but I'm not sure if we're going to get there. The safety testing so far has been quite extensive, but we're going to try, and then do some modeling at the end. I do want to acknowledge the three manufacturers here, Alga Biosciences, Blue Ocean Barns, and Symbrosia. It's not easy to come to the table and sort of say, hey, let's do some side-by-side comparisons, but that's the kind of work that's really needed. And historically in animal science, that's not the case. Typically, you work with one company and that's it, and you present the data, but you really need these kinds of studies to really push forward technology, and I think there's opportunity that we all can learn together. I thank them. They've been wonderful to work with. The first study was looking at bromoform stability, rather simple in design, but we had different temperatures, different 16 weeks of duration with sunlight or not, and then we analyzed bromoform by GCMS. We had synthetic bromoform, pelleted Asparagopsis taxiformis, and then oil-immersed Asparagopsis taxiformis. Overall, in scenarios where it's cold, right, and no light, stability looks great. That said, once you start to apply heat and light, others have shown this too, that the stability of bromoform, since it's highly volatile, becomes more questionable. One thing that we noticed that for the most part, if you keep things cold, which is what we did in our experiments, but right before treatment, you had really sort of strong stability, and we were confident that we were delivering what we thought we were delivering, at least what we were being told was the concentration of bromoform. But that said, the oil-immersed Asparagopsis taxiformis had fairly good stability over 8 weeks, regardless of the condition. In our first pilot study, we had 50 cows. We had a control, no treatment, and then 5 cows per product and per dose. And we went with 15, 22 and a half, and 30. So it's very similar to what Ermias was showing in his mean. We did that because we were trying to target a 40 to 55% reduction in methane and not trying to have a drop in feed intake. We didn't want the intake. One of our criteria moving forward to the more extensive studies was to be able to say we don't want the drop in intake, and we want to keep milk bromoform residues as low as possible, but no compromised milk production. So those are the 3 things we evaluated as our criteria to figure out how we move forward. But this was a 4-week study. We just had methane measurements before and after, relatively not intensive, just to get some quick data. What we observed was anywhere from roughly 20, 30, up to 48% reduction, depending on the product. We have oil-coated Asparagopsis on the left in yellow, the pelleted in red, and the synthetic in purple. I think they're a little bit lower than I anticipated. We did not measure bromoform in this particular study over time. We didn't measure it at one time when we received the initial product. I'll talk about that here in a minute. But for the most part, the Asparagopsis products behave very similarly. The synthetic was a little less in terms of methane reduction. In terms of milk production, this is energy-corrected milk yield. For the most part, no changes. At the higher levels where we did see a little drop in dry matter intake, we are seeing a small drop in energy-corrected milk as well. Again, more so for Asparagopsis treatments. We looked at bromoform initially, and trust me, I don't know, there's only two types of studies that create anxiety for scientists, bromoform studies, I think, and heat stress studies. I'm doing a heat stress study right now, and it's brutal. But this one, there's a lot of attention on this, and making sure that we put the cows back in the herd and things like that. But we did detect some background bromoform levels in our baseline samples, which I think is something we need to maybe learn a little bit more about in terms of what does bromoform look like in milk and tissues, just at farm level, regardless of they're being fed seaweeds or not. We did see an increase in frequency of bromoform. Statistically, there wasn't a difference in bromoform concentration in this particular study, but again, it was a pilot. When we took the treatments out at week 5, there was nothing detected. We then had a 12-week study. In that 12-week study, we had a very intensive, so again, baseline week 3 and week 12 was intensive measurements, including blood, urine and fecal collection, rumen fluid, et cetera. We had chamber work in here as well for a subset. But we ended up going with the 22.5 mg per kg dose, simply because that was a dose that didn't drop intake and didn't compromise milk production, even though there was an increase in residues, 16 cows per group. Here, there was a small difference in intake, but it was specific for the pelleted. They had about half a point difference in total dry matter intake. Energy-corrected milk yield, though, was a little bit more favorable compared to control for that treatment. And for all the oil and synthetic, there was not a reduction like we saw in the previous. I will say in this particular study, these were lower milk-producing cows than the pilot. In terms of efficiency, the pelleted had better efficiency as relative to the control. When we looked at methane production overall over the duration of the 12 weeks, there was a decrease in methane production, but it was not the desired 40% that we were sort of targeting based on our pilot data. It was much less than that. We can see here too, methane yield, there was a difference, and methane intensity, there was a difference. But again, it wasn't what we expected based on the pilot. The effects were a little lower. We look at the time course data. This was what was for us was a little bit concerning. In the pilot study, we had week 3. We had basically a 4-week study, and those measurements happened from week 3 to week 4. So again, we had about a 40% reduction, especially for the asparagopsis treatments at that short duration after 3 weeks. You can clearly see it there, but over time, things started to creep back up. We did do a ramp up the first week in this study. We can talk about ramp ups in the discussion, but it's something that okay, how do you extend that response? Because by the end, we're single digits to not being any different. Look at hydrogen, the opposite was reciprocal, so that's exactly what we would have expected, gave us more confidence right in our methane data, but there was an increase in hydrogen over the duration for all products. Wrapping up here, the bromoform was higher. We also had higher iodine in terms of concentration and yield, as we would expect. That was the case for all the products. And our conclusions here is that bromoform stability and uniformity under normal farm conditions should be prioritized moving forward, how we think more about that. Because I just got data this morning. I know we targeted 22.5 mgs, but on the oil-coated, we actually fed 20. And actually on the synthetic and the pelleted, those values were closer to 18. So the amount that we were feeding was not what we intended to. We did measure it every week. We just got that this morning. I just said these are raw averages. And so that makes me think go to the national meetings and they report bromoform levels, I don't think people have measured as much as they should be measuring it, if they're measuring it at all. And they really should be routinely measuring it because people handle it a lot differently. Background bromoform residues and tissues need to be preserved. Product differences exist. But I'm not presenting all the health data. There was really nothing in terms of health, digestibility, fermentation, really nothing going on in terms of how these products were affecting those variables. Moving forward, last slide here, looking at the effects of bromoform and fatty acids. We just enrolled these cows to do this. We're moving forward with a ramp up. We're starting out at 22.5 for a month, and then we're going to go ahead and kick that up to 30. And then for experiment 3 and 4, we are doing rumen fluid metagenomic and metatranscriptomic work at both that time where you get the 40% reduction and at the end where you've only got the 10% reduction to see what's going on and why is it different. Heather Huson's doing some DNA methylation work to see if there's some sort of host genotype interactions, even though we got really small numbers. So we have a couple small studies going in our chambers looking at the contribution of feed intake reduction to that methanogenesis drop, and hopefully we get to that dose titration study. So thanks CDFA, huge supporters. Thank you for all your help, especially because all the safety costs. Danone and Gersten were really powerful in this. Thank you.
Okay, so to complete our trilogy, please welcome Dr. Terry Engle from Colorado State University. Welcome, Terry.
Oh, there it is, hiding on me. Everybody hear me okay back there? Okay. It is a pleasure to be here, and I want to thank the committee for inviting me to talk a little bit about where we're at with our research. We are, and I'll just say this right now, we're a little bit not quite as far down the road as everybody at the two previous speakers on our research project. And I'll just say this, nobody probably in this room does this, but we worked together on a big grant. I said, sure, I can do it. It was submitted and I forgot about it. And then you get a phone call saying you got to do it. And I said I would do it a long time ago, and then everyone's high-fiving each other and I'm hiding in the corner. So at the end of the day, we had to get geared up to do kind of a quasi-semi safety study for bromoform. And so our goal was really to understand the deposition of bromoform ruminant tissues. This has been done before, but as you heard previously, that is a concern. So, and then the excretion of bromoform, is it coming out in the urine, the feces, and the one part that really is challenging is it coming out the front end of the animal. And that's what we had to develop a method to get that done. So really quick, these are the objectives of this study. And again, I want to be real careful here because we are, again, not that far down the road on this. And so the little data that I will present is preliminary data. We just finished the first phase of this study 2 Sundays ago. And so we're getting there. But these are the main objectives of or specific objectives of the study, to look at methane reduction, to understand the pharmacokinetics of bromoform. And some of these studies have been done, really good studies out of New Zealand, looking at that. Quantify the potential residues. So this is a big part of this study, is to look at parts and pieces of the animal. And then the hard part for us was to quantify the environmental contributions, both in the urine, feces, and then what comes out of the front end of the animal. So before, I am not an analytical chemist, and so I was very fortunate before we even started this study, when we started talking about it, to run over and work with our folks at the Analytical Resource Corps at Colorado State University, and they helped us to validate and verify the analysis both in gas and tissues and different matrices. And so my hat's off to them. We got a bunch of the papers that have been previously published, and they worked tirelessly for about a year to get everything set so that we could analyze what we needed to analyze. Before we started with any animals, we did some in vitro work. And this is not surprising, but we did a dose response study where we added, 0, 1, 10, 100, or 1,000 micrograms of bromoform in our in vitro system. This is a batch culture system. We still use the old school vaccine bottles. And we saw pretty much what you see in the literature, with one exception that I think is pretty interesting and probably needs to be studied in the future. But we saw the acetate propionate ratio, ammonia and methane produced per gram of dry matter digested decrease, which has been reported in the literature as dose increased. We also indicated or noticed, probably the one thing that I was really interested in is this nitrogen right here. We did see that as bromoform dose increased, the concentration of ammonia nitrogen in the rumen decreased. And that is an area that I think probably needs some further research into why that is actually happening. Another area that I wanted to make sure I understood was what happens with pressure and gas. And I wasn't challenging Henry's law or anything. I just wanted to make sure that if we let those closed in vitro bottles increase in head space pressure, could we force the bromoform gases, dibromomethane or bromoform, back into solution? And in fact, we were able to do that. And so long story short is, it's no surprise, as bromoform increased, we saw a reduction in methane production. We also were interested in bromoform and dibromomethane concentrations in the liquid component as well as the gas cap. And you can see here that as these bars right here are bromoform, this is in the liquid fraction in our in vitro system, as time goes on, it drops and we see an increase in dibromomethane, which I think makes sense. We also see the same general thing taking place over here as time goes on and the gas cap, bromoform goes down, dibromomethane goes up. And so that's the area that got us pretty interested in trying to figure out what's coming out of the front end of this animal. And so I think a lot of you in here have built head boxes before. And I did have the privilege of working with Don Johnson while he was still alive at CSU, and worked with some of his chambers. And so we built several head boxes and did all the calibrations, the ethanol burn test, and I felt pretty comfortable measuring CO2 and methane and oxygen. But we needed to figure a way to concentrate the bromoform that we give to these animals. And so we did a lot of weird things. We put bromoform inside our head boxes. We ran the head boxes. We ran some in vitro fermentation systems in the head boxes to try to quantify and capture the bromoform or the dehalogenated compounds in our head box system. And so, we also use our cannulated animals. We found the biggest one we could, about 1,300 kilograms, and we put a bunch of bromoform in, and we're able to capture what's coming out the front end. So the challenge here is really the concentrations that we're working at. And so what we have done and fitted our head boxes with these sorbent tubes, both on the intake and the output. And it took a while to figure out. So these are basically SPME fiber tubes on steroids. It's a big one. And we capture the bromoform as it's coming out, or dibromomethane. We measure what's going in too, so we can adjust for that. And so it took us a while to calibrate our systems, but at the end of the day, we were getting some pretty good recoveries. Okay, and so that said, what is the experimental design? And so what we've put together here is we're studying beef and dairy cattle. We have 3 independent phases. We just completed phase 1. We have 12 animals per phase per breed. And then the doses, we only use 1 dose in here, and that was 1 milligram bromoform per kilogram live weight. And so that's a pretty hefty dose based off of some previous literature. And so the first experiment was basically an oral gavage and measuring what's going on with bromoform and its metabolites over a 24-hour period. And we just, like I said, we just finished this one up. We also put jugular catheters in these animals to look at how much shows up in the blood over time. And so they stayed in these head boxes for 24 hours. And we measured the blood, urine, fecal collection. We put diapers on these animals, urine harnesses, all that good stuff. And then we harvested these animals at the end of the 24-hour period. So preliminary results, and these are preliminary. We did see a pretty hefty reduction in methane, which we expected to see. Total hydrogen excretion increased. So we're measuring that as it's coming out the front end of the animal by about 200%. 24-hour dry matter digestibility was similar across treatments. It's not the right way to do a dry matter digestibility study, but we plan on doing that in the future with the longer-term dosing of bromoform. We didn't notice any, upon necropsy, no gross abnormalities in any tissues. We're still working on the histology work. We did do apparent nutrient absorption and retention. That was not different. And we did, we measured every part and piece of that animal. The only ones we have back right now are the liver, the lungs, muscle, and kidney. And we did not see any accumulation of bromoform or any of the halogenated compounds in those tissues. And I'll highlight the lungs here in a minute. And then when we look at what shows up in the blood, this is a rough indicator of what happens. So this is bromoform, that big peak, dibromomethane. And then what shows up in the urine, not so much bromoform, but dibromomethane shows up in the urine And so what we're doing is trying to quantify exactly how much we give and how much comes out at each area of the animal. And so long story short is, if we look at this animal and we look at the total bromoform or the dehalogenated compounds that we can account for, about 20% of that bromoform is going to come out the front end of the animal, and about 40% of the dibromomethane we were capturing coming out the front end. And so what we still have to do is figure out how much is coming out in the fecal material, as well as the urine in milk. And so we're not quite there yet. So moving on to the next two phases that we have to do, basically we're going to feed bolus dose bromoform, we're going to feed it in meals for 28 days, and then do these measurements every 7 days. And at the end, we'll euthanize those animals, and then a longer term feeding in 48 days. And so with our little sorbent tubes, we have added to the mess here, and we've identified 252 unique compounds that are coming out of the front end of that animal that are associated with the study design. And so we're in the process of trying to separate out control animals and bromoform treated animals. So with that, I will go ahead and wrap up and say thanks to this big group. This is my lab group, and they are fantastic, and I put them through a lot of hell.
So can I ask our two other speakers to come and join the panel, please? Thanks. Just getting my equipment up and running. Okay, awesome. Okay, thank you all for some great presentations and getting us up on the latest and greatest. To sort of warm you up, we didn't hear much about the mechanism of how bromoform works. I know there's work in Germany that shows that, you know, it does affect not just methanogens, but also acetogens, who are, you know, quite important when it comes to how the rumen functions. What do you know about mechanisms? Can anybody from the panel comment on that?
Okay, I think it works. We haven't really done a lot on the mechanism part of that, and I know there's some work in the literature that shows how bromoform is affecting and reducing emissions, and a lot of the literature points towards its effect on the vitamin B12 concentrations, and how towards the end. In which is catalyzed by enzymes and interfere on the enzyme side of things. So it's kind of similar to how 3-NOP works, but not quite the same. I think 3-NOP is more specific, while the bromoform is wider in its effect.
Do you think it's still a gap?
Yeah, I mean, yes, I think there's a gap to try to understand a bit more on that.
Yep. Joe?
Yeah, I think I think we need to consider alternatives that besides just bromoform itself sometimes. I mean, I don't study the literature maybe as much as I should in terms of other ingredients, other compounds that are within Asparagopsis, for example. I know there's been some work in this as well, trying to highlight what Asparagopsis can do beyond bromoform in terms of reducing methane. And there's other alternative halogenated compounds that could be potentially be in there. There's also non-halogenated sort of like phenolic compounds that might also have some sort of contribution that we don't know enough about. I only know this from my own perspective, where I had equal intake, bromoform intake, equivalent across the synthetic and an Asparagopsis, yet the intakes, the methane drop was a little bit lower in the Asparagopsis, partly due to the drop in intake. So the intake part also needs to be part of the conversation.
Yeah, just to add to that, I mean, there is an in vitro study that showed that maybe bromoform is half of the story, but then you see synthetic bromoform working so well, it's like, okay, yeah, it's very complicated.
Yeah, we've got a question here, and it's aimed at Joe, but I think others can maybe give their opinion on that. It said, Joe, are you hypothesizing that an adaptation of the rumen microbiome is happening, and hence your observation of decreasing efficacy of bromoform over time?
I don't know. I was really into that. I mean, like last short 2 years ago to my life, I was like, oh, microbial adaptation. Everybody was talking about it. Then I was learning about stability. Then I was on the stability bandwagon. Maybe this is a stability issue, but I'm not. It's not a stability issue, in my opinion. I think there are some issues regarding stability that need to be paid attention to. But in terms of my approach, it wasn't necessarily a concern. Like they were being fed, even though a little bit less than the desired, it was consistent. I know that there's some work in terms of the short duration studies, something that we've even seen too, that I haven't published in terms of the short-term ramp up and how that can be beneficial. So yes, I think that needs to be considered. We're working with Christopher Mason. He's a well-known microbiologist to be able to figure out maybe what's going on microbially at week 3 versus week 12. I'm really happy we chose those sampling times because it really helped.
Yeah, I think in terms of adaptation, microbiome adaptation, we haven't really seen a lot of evidence to that. We did this work, 5 months work, 2018, 2019, in beef cattle. We didn't see any drop-off. And the current one that we're doing, almost finishing now, through the Ted Odysseus project, is that 18 months, 20 months, there hasn't been any drop-off. So these are animals that were fed Bromoform right from they were weaned 2 months all the way to 18, 20 months. We have not seen any drop-off. So we've seen like 50% reduction up to, until the day they're harvested.
And what's the feeding level there in that study?
It's about 32 milligrams per kilogram. So we're trying to get up to that 55% reduction rate. But the good thing with this is that we are taking samples, microbiome samples, and doing deep sequencing every month on all of those 24 animals there. So I don't have the data right now, but this has been analyzed at UC Berkeley. And so we will have a very detailed analysis of the microbiome so that we understand what's going on right from the start when we put the bromoform and which microbes are being sort of inhibited and which ones are promoted by inhibition of the methanogenesis. So we will have this very rich data soon. But the evidence in terms of just looking at the methane is that we haven't seen a drop-off after such a long time.
Great, thanks. We've got a couple of questions and I'll kind of bowl them into one. And it's really around, safety, particularly bromoform, talk about a human carcinogen. What is the portfolio of evidence that we're going to need to have to give confidence to, both the regulators, but also, at the end of the day, farmers as well. We've got to learn from the story. We're all aware of it, what happened in Denmark with 3-NOP. So yeah, maybe let's talk a little bit about, and then maybe Terry, we'll start with you because you've obviously starting to do some of that safety studies that's going to be needed for regulatory.
Yeah, obviously tissue concentrations are the most important, I think. And one of the things that I think we need to look at too is that animal eructates over time. Does that bromoform come back in to the lungs? And so I would say if we need to spend a little bit of time talking or looking at the lung tissue concentrations, the headbox system that we use will pull that gas away. So over the next phases of our study, they will be consuming meals of bromoform for 28 days, and then we'll be able to see if it's showing up in the lungs. One thing that I didn't mention here is the F word, the fetus. That's always a tricky one, and we have some data that would suggest that it's not getting into the fetus of gravid animals, and so that's I think a good thing. But the duration, we're in short duration right now, and so I don't know what the long-term effect is. So that would be, I think, an important thing to look at.
Before we go to the other panel, there's a top question coming up for you, Terry. Do you think that 20% of bromoform excreted is driven by how you fed the bromoform?
100%.
Do you think this will change when it's delivered in a feed and consumed throughout the day?
100%. And again, we're just starting out on this. Obviously, if it takes a little bit longer to be digested in the rumen, it'll stick around a little bit longer. That would be future experiments. But what we're really trying to do is do an accounting of the bromoform, and if we can capture where's it going, and relative to the dose that we gave, how much can we understand, quantify where it went, and so a lot of it's coming out the front end in the way we were doing it in this experiment.
Yeah, so just going back to that higher level question, what is the portfolio of evidence that we're going to need in this space. I think somebody had a question, I was trying to find it, around, when is academia going to stop just doing the feed 'em and weigh 'em type trials and move on to the type of work that is going to give confidence to businesses to be able to use this type of technology? What do we need to be doing?
Yeah, I think Terry alluded to it. I mean, he's doing the work. I think the residue analysis is going to be very, very important because to make sure that there is no unintended consequences. So, yeah, I think we feel pretty comfortable on the efficacy side of things, but on the residue, I think that's really going to be one of the most important things. And this is what we're doing in this experiment, is that after you feed for so long, I mean, a lot of the work has been done in a short period, but if you do it for a long period enough, where that is what we wanted to happen in the commercial sense. So what happens if there is any residues there? And so that work needs to be done. And some of the work has been done already. I think, I don't know if Ranka is around here, who's done some work on residue in milk. So I think those kind of work we really need to build up and make sure that there are no unintended consequences.
Yeah, I need to say something about this. We don't have a lot of data, but we did collect on the last study. We did a subset like this. Did some biopsy work. So we had fat biopsies, liver biopsies, and muscle biopsies. But I want to make a point on that very first study that we did with a pilot. We had an animal that unfortunately she tripped, hurt her leg, and she had to get euthanized. So we used that opportunity to do some total tissue collection, look for bromoform. And the concentrations in fat tissue were crazy high in this animal. And we went back, she was on a low dose for about 3 weeks. We went back and looked at her baseline milk bromoform levels of the 50 cows. She was number 50 in terms of having the highest milk bromoform levels at her baseline before she even started treatment. So I don't want to be pointing like I don't want you to come to me like in 5 years and say, hey, Joe, you didn't tell me that there's potentially bromoform and other sources on herds and farms. I think it's something we just need to pay attention to. I don't think it's a huge issue from my perspective, but it's something we observed.
I want to, there's one or two questions I'm going to pull into one, and it's really around productivity, right? We, what are we actually seeing? When you're talking, there's a lot of people out there pitching methane mitigation plus a productivity co-benefit. Obviously, if you're doing anything with productivity, if you don't see greater hundreds of animals in your trial, then it's very difficult to talk about productivity. But you're feeling from what you're seeing in the work to date around productivity and potential co-benefits. And Ermias, you've obviously seen potential, some positives and some negatives. So do you want to talk on that?
Yeah, so I think in terms of productivity for dairy, it's really related to intake. So if you got your intake right, and looks like a lot of these things have been solved, and the intake is kind of stabilizing. I think Joe, you showed that the intake was okay. Yeah. So I think with the productivity with the intake on the dairy, but on the beef side of things, the issue we have is that the changes are small, so that we need a lot of animals to be able to say confidently that there is an improvement in productivity or not. What some studies have shown, in Australia, they've shown an increase in productivity towards the end of the sort of the feedlot period in beef cattle. Some did not see, and here at UC Davis we've seen before, and the study we're doing right now, we're starting to see that improvement as well.
So there's a question on that actually. What is driving the increase in the ADG, and why is it only observed in the finishing period? Got the answer?
Yeah, well, it's a good question. I mean, this is what we're trying to figure out. I mean, theoretically, if you're reducing methane, you're saving some of the carbon, although you're still losing hydrogen, and that saving carbon might be converted into the tissue. But why right at the end? Well, why not throughout? So this is what we need to figure out. And the evidence that I've seen from literature in Australia and here in Davis also is the last 2-3 months that we see the changes. So why are we not seeing sort of throughout the whole lifetime, and why is it concentrated on the last one?
Yeah. For Terry, there's a couple of questions around ozone depleting substances and being eructated out. Obviously, bromoform is its closely related cousin, is an ozone depleting substance banned under the Montreal Protocol. So yeah, thoughts on that?
Yeah, I did open a can of worms with that, with all those compounds coming out of the food of the animal. I knew that was coming. And so we don't have them all identified yet, but there will be some things, hydrogen sulfides, those types of things that we know will show up. And I think we just need to be careful on trying to quantify those. We don't have standards right now for those components, but for what we've seen so far and the limited data that we have, we're getting quite a bit of bromoform coming back out. Again, this is bromoform and propylene glycol, so it's coming out pretty quick. Within 8 hours, it's depleted.
Yeah. Cool. Thanks. Joe, there's a top question for you. If the cows are on the same water source, how do the controls go from non-detectable bromoform to detectable by different weeks? Is there a mix-up in feed, was the question.
I'm assuming you're referring to the pilot study table that week 5 and week 6, they were taken off treatment. So there are things that happen from baseline to the 4-week experimental period to the 2 weeks post-experimental period in terms of dietary changes, even a pen change from baseline to the experimental period. But that, if you're referring to the not detected in weeks 5 and 6, they were off treatment at that time. So we just wanted to show that you take them off treatment, everything should go back down to 0. My concern there was at the baseline, we had some animals, like 8 animals out of 50, that had a positive bromoform test. We did try to go back. We didn't discover this until several months later. So we collected different types of feed samples. We took water samples from the pens they were in and the new pens, and those didn't, we don't have all the data back on that, but the water wasn't the source.
You know, we are researchers up here, but there is a question here. I'm not sure if anybody can take it. What is the expected cost to producers? So it's that question: who's gonna pay, particularly in the long term? Anybody got some opinions or thoughts on that?
I think in terms of the cost of the additive itself, I think for the bromoform base, we don't know. At least I don't know any number that's been thrown out. But it has to be obviously competitive with whatever else is happening there. But I mean, there are mechanisms for which how we can pay. Well, one is through the carbon credits. So the carbon credit system in the US is definitely ramping up, and I see a team from Athens here who are doing this work where you can use additives to, first of all, you have to come with a protocol, but after that, then if you're able to account for a reduction, then you'll be able to get some credits, and you'll be able to sell the credits as well.
Yeah. A question around, obviously, I went back and we talked about Lanigan's study. He did a 100% knockdown over 24 hours. Is there a tipping point for bromoform-based approaches? Is there, a point where if you push it too hard, you'll topple the system? And is there an area where we should be targeting? Obviously people can get up to 90% reductions over short term, but do you have a feel for what that tipping point might be, or the upper limit? And does that differ between beef and dairy?
Yeah, I would, on the in vitro side of things, we did take those doses super high to where we stopped fermentation. So we can get to a situation where, it is inhibitory for microbial growth. But as far as, do we need to be at 50% inhibition and not have impacts on growth performance? I don't know the answer to that question.
Okay, cool. This is a question you should never ask a scientist. If you had budget and capacity, what trial do you wish you could run? But we've got, I think, I never wore my glasses, but I think it's about 2 minutes. Yeah, maybe let's go through the panel. Your wish list. Let's tell the audience there could be some interesting people out there listening who have a big wallet.
Yeah, I mean, I'll start. I mean, this is something we talked about last year, and we talked about it throughout the year in different ways. I mean, it's obviously unrelated to what I do directly, but communication is important. I think these types of technologies with this kind of risk, and we're talking about it extensively the last hour, is something that it's not like going to be ignored, okay? Especially if this is something that resonates to a herd system. So how do you communicate technologies like this, the importance of it? If it's something we're going to keep pushing, which we obviously are, we need to understand consumers and how they're going to respond to these things, and how do we better educate. And we have put thought into that to try to make that, to do that survey work. And and develop a communication plan.
Cool. Thanks, Joe. Miss.
Yeah, well, I guess we talked about the productivity issue and how do you solve that in having a lot of animals. So if I can work in a feedlot with 1,000 animals and be able to measure their daily gain every day, for the entirety, I think that would be great. But I don't think that will ever happen.
Actually, I think there's a trial in New Zealand ongoing that's doing just that. So keep an eye out for Chief. And
I would just say that what Joe mentioned earlier is what is the background information or bromoform concentrations or their dehalogenated compounds, because that I think will help with the approval process. If we know that they're already there, then I think we would have a better understanding of what is accumulating in that animal because of what we're doing feeding wise.
Well, okay, so the story still continues. We haven't finished the book, but maybe next year when we come back, there'll be another chapter. So just thank you to all the audience members for their questions, but also to the 3 panel members. I hope you all feel that you've got the state of play when it comes to research in bromoform. Yeah, thank you.